Good morning to everyone and welcome in this lecture of GATE examination. First upon, I really congratulate to you all for applying GATE examination 2025, taking right time, right decision. You have lot of opportunities in various field, but after qualifying this GATE examinations, have a opportunity that is as a junior research fellowship, as a MTech, also provide fellowship. But GATE examiners, this path is not so easy. You have to work hard and honestly to achieve your goal. So welcome. Let's started our today's lecture, that is GATE examination expected multiple choice questions life science subject. So already we'll previously discussed, that is what is GATE, who is eligible for this exam, BPharm is eligible or not, pattern of examination, syllabus, total marks. Total marks is 100 expected multiple choice questions. So GATE is a graduate aptitude test in engineering, is a national level examination. Eligibility of GATE, final year of their undergraduate degree, BTech, BE, BSc, BPharm or equivalent are eligible to appear for GATE. And is BPharm is eligible? Yes, BPharm student also eligible. Pattern of examinations. This exam is consist of multiple choice questions. Time is 3 hours. Opportunities of GATE qualified students. Valid GATE score can be used for seeking admission to master programs. Also doctoral programs relevant branches. Financial assistance 12,400 for MTech post graduation per month, and generally paid for 22 months PG scholarship, GATE equivalent to GPAT. Also financial assistance given for PhD programs, direct PhD after BE, BTech, MSc equivalent or with valid GATE score. This is also opportunity to entry direct PhD after BE or BTech or MSc equivalent with the valid GATE score. Stipend is that RS 37,000 per month for the first 2 years and RS 42,000 per month from the third to fifth year. So also private sectors also prepare the candidate, that is those have GATE qualified. So this is your good opportunity. You also visit more official website http:/gate 2025 iitr.ac.in. Marking scheme of GATE examination. The question paper is that is carried 1 mark and 2 mark questions. Also negative marking. For wrong answer choose in MCQ, there will be the negative marking. For 1 mark questions, 1/3 marking. Likewise, for 2 marks MCQ, 2/3 mark will be deducted for wrong answer. An exam date of life science subject is that Sunday 16th February 2025. You have to remember the date. Subject life science. Life science in that chemistry is a compulsory subject, 25 marks, and optional 30 marks, that is biochemistry, microbiology, botany, zoology, food technology. You have to choice out of 5 two subjects in the examinations: biochemistry or microbiology, botany or zoology, food technology or zoology. Whatever your choice. So choice is yours. So 25 marks chemistry, 30 marks biochemistry, microbiology also 30, means the total that is 60 marks, means total optional subject have 60 marks, and chemistry compulsory that is 25, total 85 marks, and a general aptitude also 15 marks, total 100 marks paper. Mode of examination computer based test, and time duration that is 3 hours. So you have to remember this. Syllabus, next point, important point of view. Just understand any examination want to clear that know about syllabus. First, that is the role, that is to understand the syllabus, prepare the notes, solve the question paper. That is a key strategy of examination. So syllabus, that is first atomic structure and periodicity, second section structure and bonding. First unit in that section 1 in that Bohr's model, quantum mechanical model, structure bonding, ionic covalent bonding. So this is the syllabus of chemistry. Then section 3 sp and d block element, chemical equilibria, electrochemistry, reaction kinetics, thermodynamics, structure, reactivity, correlation and organic reaction mechanism. And section 9 chemistry of biomolecules, amino acids, proteins, nucleic acid and nucleotides, peptides and sequencing by chemical enzymatic proteolytic methods, Beers-Lambert's law. So this is the chemistry syllabus. Next, optional subject biochemistry. Looking to the syllabus, section 1 organization of life, importance of water, structure and function of biomolecules, amino acids, carbohydrate, lipids, protein, nucleic acid, protein structure, primary, secondary, tertiary and quaternary, enzymatic kinetics, regulation, inhibition, vitamins and coenzymes, bioenergetics and metabolism, TCA cycle, biomolecule separation techniques, ion exchange, signs exclusion and affinity chromatography, cell structure and organize, biological membrane action potential. Section 5 DNA replication, transcription, translation, DNA damage and repair, biochemical regulation of gene expression, RDNA technology and application, polymerase chain reaction, immunity system, innate and adaptive. Innate means inborn natural immunity to protection of foreign particles. Cell receptor, beta cell, T cell activation, major histocompatibility complex to identification of self and non-self molecule. Immunological techniques, immunodiffusion, immunoelectrophoresis, RIA, radioimmunoassay, ELISA, enzyme linked immunosorbent assay, identification of AIDS. Monoclonal antibodies, rituximab for treatment of cancer, anti-cancer drug, rituximab, autoimmune disorders treatment also, and their applications. So this is the syllabus of biochemistry. Next, second optional subject microbiology. So microbiology in that historical perspective, methods in microbiology, microbial taxonomy and diversity, prokaryotic cells structure and function, microbial growth, control of microorganism, microbial metabolism, microbial disease and host pathogen interaction, chemotherapy or antibiotics. Antibiotics, the agent with substance which inhibit the growth of microorganism. General characteristics, antimicrobial drugs such as penicillin, tryptomycin, sulfonamide, classification, molecular mechanism, microbial genetics, microbial ecology. So this is the syllabus of microbiology. Next, last subject, that is 15 marks, verbal aptitude, general aptitude syllabus, verbal aptitude. In that, basic English grammar, tense, present tense, past tense, future tense, articles, a, and the, where is uses, adjective, prepositions, conjunction, verbs, noun, basic vocabulary, Adams and Pierce, reading comprehension, quantitative aptitude, analytical aptitude, logic, deduction and induction, spatial aptitude, transformation of shape, etc. So this is the first we looking the syllabus. Next we turn to the questions, multiple choice questions. So one by one subject. So you have to remember chemistry 25 marks. Then optional subject, that is 30 marks. Biochemistry, microbiology, botany, zoology and food technology. Out of that you have to choice is yours. So first question, last year GATE examination asked two questions. The correct order of electronegativity. The correct order of electronegativity is option E, aluminum is greater than silicon, greater than phosphorus, greater than sulfur. B, aluminum is greater than sulfur, greater than silicon, greater than phosphorus. C, sulfur is greater than silicon, greater than aluminum, greater than phosphorus. And D, sulfur is greater than phosphorus, greater than silicon, greater than aluminum. Electronegativity means an atom's of ability to attract shared electron itself. Electronegativity means an atom ability to attract shared electron itself. Also it measures relative strength and element ability accept electron. So answer is D. Next, the molecule with non-zero dipole moment. So dipole moment measure separation of positive and negative electron within system. So option correct answer is that NO, nitrous oxide, and SO2. Next, upon addition of compound X to an aqueous AgNO3 solution, a white precipitate appear instantly. Also, X does not have exhibit geometrical isomerism. The correct option is yes for X is R. Option A, option B, option C, and option D. So B and C is the correct answer. Next, the paramagnetic species among the following is are given atomic number. Option is given option A, option B, option C, and option D. So A, B, and D is the correct answer. And fifth, an enzyme catalyzed conversion of substance at 219 Kelvin proceed by Michael-Menten mechanism. The Lineweaver-Burke plot for the analysis of experimental data has an intercept along the y-axis of 0.357. Which of the following correct answer? A, 5.88 mmol, B, 5.88, C, 2.80, and D, 2.80. Option A is the correct. Next, unit wise the question will discuss. Which of the following postulate is a part of Planck's quantum theory? A, energy is quantized and can only exist in discrete packed packets called quanta. B. Energy is continuous and very smoothly. Light behave only as a particle and the frequency of radiation is inversely proportional to its wavelength. So option A is the correct answer. Next, which principle explain why number of two electrons in an atom can have identical quantum number? Option A, Paul exclusion principle. Option B, Hund's rule. C, Afobe principle. D, Heisenberg uncertainty principle. So option E is the correct answer. Which of the following elements has highest first ionization energy? Option A, sodium. Option B, magnesium. Option C, oxygen. Option D, neon. So answer is neon. Next, which periodic property decreases across a period and increases down a group? A, electronegativity. Atomic size. Ionization energy. Electron affinity. So atomic size. Which periodic property decreases across the period and increases down a group? Next, structure and bonding. Which of the following bonds type are characterized by sharing of electron pairs between an atom? Option A, ionic bond. Option B, covalent bond. Option C, metallic bond. And option D, hydrogen bond. Covalent bond characterized by sharing electron pairs between the atoms. Next question, according to VSEPR theory, what is the shape of molecule with 4 bonding pairs and 0 lone pairs on the central atom? A, linear. B, trigonal, planar. C, tetrahedral. And D, octahedral. Tetrahedral. Next, what is the bond order of molecule in which there are 4 bonding electrons and 2 antibonding electrons? Option A, 0. Option B, 1. Option C, 2. Option D, 3. And answer is that B is the correct answer. Next, which of the following molecule have permanent dipole moment? Permanent dipole moment. CO2, carbon dioxide, CH4, water, H2O, O2. So water have permanent dipole moment. So it measures separation of positive and negative electron. Next, which of the following is a common characteristics of transition metals? A, they have low melting points. B, they exhibit variable oxidation state. They form ionic compounds only and they do not form coordination complex. B is the correct answer. They exhibit variables oxidation state. Next question. In coordination complex, the ligand forms bond with metal ion through which type of orbital interactions? A, sigma bonding. B, pi bonding. D, orbital interactions. And D, both sigma and pi bonding. Orbital interaction. Next question, which of the following compounds will not exhibit magnetic properties? Answer D is the correct. Next, what is the coordination number of central metal ion in octahedral complex? Option A 2, option B 4, option C 6 and D is 8. Correct answer is that C 6. Next, which of the following is true for solution at equilibrium? A, the concentration of reactant and product are equal. The rate of forward reaction equal. The rate of reverse reaction. C, the reaction has stopped. And D, the equilibrium constant is 0. Question is that which of the following true for solution at equilibrium? So when the rate of forward reaction equal to rate of reverse reaction, that time the solution at equilibrium. So B is the correct answer. Next, second, which of the following is the correct expression for the solubility product KSP of salt? So A is the correct answer. Next, the common effect result in the dash dash of the solubility of salt in the presence of common ion. Common ion effect result in option A increase, option B decrease, C no change, and D complete dissolution. So decrease, common ion effect result in decrease of the solubility of salt in the presence of common ion. So once solubility is decreases, then it affect the pharmacological actions, means decrease the surface area and decrease the pharmacological actions also. Next, what is the pH of 1 into 10 raised to 6 molar HCl solution? Option A 5, B 6, C 7, and D 8. So 6 is the pH. Next, electrochemistry. According to Nernst equation, which factor does not affect the cell potential? Option A temperature, option B concentration of ions, C pressure, and D mass of the electrode. So answer is correct, mass of electrode. Next, what is the main implication of Coriolis law independent migration of ions? Option A, the conductivity of solution depends on the ions present. B, the total conductivity of solution in the sum of conductivities of individual ions. C, conductivity increases as the concentration of ions increases. And D, conductivity is independent of the concentration. So B is the correct answer. Next question, what is the standard electrode potential for reaction zinc 2 plus 2 electron minus zinc sulfur? Option A, option C is the correct. Next, EMF of cell 1 volt at 25 degrees Celsius. What is the value of equilibrium constant? One into 10 raised to 5. Reaction kinetics. Kinetics means rate, moment. The rate constant of reaction has unit mole per liter raised to 1. Second 1. What is the order of reaction? Second order is the correct answer. Next, for first order reactions, the half life independent of concentration of the reactant. Which of the following true for a first order reaction? First order reaction, the half life is independent of concentration of reactant. So C is the correct answer. The rate of the reaction decreases with time. Next, which of the following factors does not affect the rate of elementary reaction? A, activation energy, B, temperature, C, concentration of reactant, and D, solvent used. So answer is D, the solvent used. Next, which of the following characteristics of enzyme catalyzed reaction? Enzyme catalyzed reaction. A, they increase the activation of energy of the reaction. B, they decrease the activation of energy by providing the alternative reaction pathway. C, they increase the concentration of reactant. They do not follow the Michael-Menten kinetics. So B is the correct answer. They decrease the activation energy by providing the alternative reaction pathway. Next, thermodynamic. Which of the following is true about a reversible process? A, an ideal process that can be exactly reversed. B, the system is always at equilibrium. And C, the process occurs infinity fast. And D, it always involves constant temperature. So true about reversible process, it is an ideal process that can be exactly reversed. Next, the first law of thermodynamics state the total energy of system and its surrounding is constant. What is the mathematical expression of this law? Means energy neither created nor be destroyed but converted into one form to another form. So delta U equal to delta Q minus W is the mathematical expression of this law. Which of the following is correct for irreversible process? A. It is always at equilibrium. B. It is characterized by gradual increase in entropy. C. It occurs with no energy loss. D. It does not involve any change in entropy. B is the correct answer. It is characterized by gradual increase in entropy. Next, Gibbs-Helmholtz reaction. Delta G equal to delta H minus delta T. Yes, what does the delta G represent? Option A, entropy change of system. B, heat absorbed or released at constant pressure. C, free energy change at constant temperature. And D, work done by the system. So free energy change at constant temperature is the correct answer. Next, structure, reactivity, correlation and organic reaction mechanism. First, which of the following true for SN2 reaction mechanism? The rate depend on the concentration of nucleophiles and electrophiles. B, the reaction proceed via carbocation intermediate. C, reaction characterized by slow rate determining step. And D, the stereochemistry of the product is the same as the reactant. So answer is the rate dependent concentration of nucleophiles and electrophiles. Next, even elimination reaction. Which of the following is the correct order of event? A is the correct answer. Next, which of the following factors affecting stability of carbocations? A, hyperconjugation. B, inductive effect. C, resonance. And D, size of carbocation. So size of carbocation cannot be affect stability of carbocations. Next, which rule govern the formation of major product elimination reaction E1 and E2 reaction? Markovnikov's rule. A, B, Setti's rule. C, Hofmann's rule. And D, Le-Chatelier principle. So B is the Setti's rule is major product elimination. Next, chemistry of biomolecules. Which of the following techniques is commonly used for purification proteins based on their charge? A, gel filtration chromatography. Ion exchange chromatography. So chromatography is a techniques stationary phase and mobile phase used for separation of alkaloids, flavonoids. So it is a separation techniques like filtration, also separation techniques. So chromatography, the question is that which of the following techniques commonly used for purification of proteins based on their charge? A, gel filtration chromatography. Ion exchange chromatography. C, affinity chromatography. And D, thin layer chromatography. So based on charge, ion exchange chromatography is used for. Also it is ion exchange chromatography used for water purification. Next, which of the following is true about Beer's-Lambert's law? A, it describes the relationship between absorbance and concentration of solute. It only apply the non-absorbing solute. C, it applicable only at high concentration of solute. And D, it relate absorbance to the temperature of the solution. So A, it describes the relationship between absorbance and concentration of solute. Next, which of the following not a function of lipids in biological system? A, energy storage. B, membrane structure. C, enzyme catalysis. And D, hormonal regulation. So enzyme catalysis, lipid in biological system. So lipid is not energy storage. Lipid have highest energy as compared to carbohydrate. Membrane structure is hormone regulations, but enzyme catalysis is not. Next, which of the following statement true about carbohydrate up to hexose? The D and L configuration of hexose are based on the orientation of hydroxyl group on the highest number of chiral center. Next, we'll turn to the biochemistry. So previous year asked a question, which of the following pairs of amino acid is not incorporated in polypeptide chain? Polypeptide chain. Option A, 4-hydroxyproline and Y-carboxyglutamate. B, carboxyglutamate and desmosine, ornithine and citrulline C, and 4 hydroxyproline and 5 hydroxylysine. So correct answer is ornithine and citrulline. Next, mammalian cell cultured low temperature 25 to 30 degrees Celsius leads to increased sterol content in the membrane. Elevated sterol in the membrane result in A. An enhancement in membrane fluidity, B. Stabilization of membrane proteins, C. An increase in membrane permeability to water, D. Decrease in membrane fluidity. An enhancement in membrane fluidity. Next, which of the following metabolic intermediate is common to glycolysis, nucleotide synthesis and glycogen synthesis? A. Citrate, B. Oxalic acetate, C. Glucose 6 phosphate, and D. Glycerol 3 phosphate. So question is that which of the following metabolic intermediate is a common glycolysis, nucleotide synthesis and glycogen synthesis, that is glucose 6 phosphate. Next, question number 4. In mammalians, hematopoietic stem cell that gives rise to the different types of blood cells are known as A. Totipotent stem cell, B. Pluripotent stem cell, C. Myeloid progenitor cell, and D. Lymphonoids progenitor cell. So B is the correct answer. Next, which one or more of the following statements correctly describe the addition of N nucleotide during the rearrangement of immunoglobulin heavy chain encoding gene? A. Addition of N nucleotide is a template encoded. B. N nucleotide are added by terminal deoxynucleotide transferase. C. Added N nucleotides are common in P and D, and D. J junction. D. N nucleotides are added by DNA polymerase. So B and C is the correct. Next, unit wise, section 1, organization of life, structure, functions of biomolecules. First question, which of the following molecules is the primary source of energy in living organism? Primary source of energy living organism: A. Proteins, B. Carbohydrates, C. Lipids, and D. Nucleic acid. So carbohydrate is the primary source. So proteins also source and lipids also source, but primary source, that is carbohydrate. Next, which amino acid is involved in the formation of disulfide bonds in proteins? A. Lysine, B. Cysteine, C. Glutamate, and D. Valine. So cysteine is a disulfide bond in proteins. Next, what is the primary structure of protein? A. The sequence of amino acids is polypeptide chain. B. Three-dimensional folding of the polypeptide chain. C. The interaction of multiple polypeptide chains. D. The hydrogen bonding between atoms in the peptide backbone. The sequence of amino acids in a polypeptide chain. Next, which of the following proteins is responsible for transport of oxygen in blood? A. Myoglobin, B. Lysozyme, C. Hemoglobin, and C. Chymotrypsin. So following protein is responsible for transport of oxygen in blood. So C is the correct answer, hemoglobin. Next, which of the following molecules are composed of glycerol, fatty acid, and sometimes phosphate group? A. Proteins, B. Carbohydrates, C. Lipids, and D. Nucleic acid. So lipids. Next, enzyme kinetics and regulation and inhibitions. Which of the following primary function of ATP in cellular metabolism? Provide the energy of cellular processes. So B is the correct answer. Next, which enzyme is responsible for catalyze rate limiting step in glycolysis? So phosphofructokinase. Next, in TCA cycle, which molecule is produced during the decarboxylation of isocitrate CO2? Next, which of the following is the key regulatory enzymes in the pentose phosphate pathway, that is glucose 6 phosphate dehydrogenase. Next, what is the primary role of nitrogen fixation in nitrogen metabolism, that is convert atmospheric nitrogen into usable form of plants. Biochemical separation techniques. Question that which chromatographic techniques separate biomolecules based on their size. So last question, that is part charge, that is ion exchange chromatography. Here questions as size. So ion exchange chromatography is cancelled option. Affinity chromatography, size exclusion, and high performance liquid chromatography. So see, size exclusion question is asked, that is based on their size. So size exclusion chromatography is the correct answer. Next, which techniques is most suitable for determining molecular weight of proteins? Molecular weight. So mass spectroscopy determine the molecular weight. UV visible spectroscopy determines saturated and unsaturated compounds. Gel electrophoresis and enzyme linked immunosorbent as ELISA is identification of AIDS HIV first screening test. So B is the correct answer. Next, SDS page electrophoresis proteins are separated primarily based on their A charge, B shape, C size, and D function. So based upon size. Next, which of the following is used for protein interaction studies? A gel filtration chromatography, B affinity chromatography, C protein interaction assay, and D both B and C. So answer is D both B and C. Next, which techniques is used for separating biomolecule based on their charge? Which techniques is used for separating a biomolecule based on the charge, that is ion exchange chromatography? Answer is B is the correct. Cell structure and signal transduction. Which of the following organelles is responsible for protein synthesis in cell? A mitochondria, B ribosome, C lysosome, and D Golgi apparatus. Protein synthesis in cell. So ribosome is the correct answer. Mitochondria. Lysosome is enzyme. Golgi apparatus. Next, which of the following is not a function of biological membrane? A separation of cellular compartment, B transport of ions and molecules, C production of ATP, D communications with extracellular environment. Production of ATP, not a function of biological membrane. Next, what is the main function of neurotransmitter in the nervous system? Neurotransmitters like acetylcholine, dopamine, serotonin, these are the neurotransmitter. Glutamate, glycine. Question is that what is the main function of neurotransmitter in the nervous system? A to synthesize proteins, B to transmit electrical signals across the synapse, C to maintain cellular integrity, D store energy. So A, C, D is not correct answer. B is the correct answer. To transmit the electrical signals to across synapse, because the imbalance of neurotransmitter result in disorders like depression, like Parkinsonism, like mania, psychosis, etc. Next, which of the following is an example of passive transport across biological membrane? Passive transport. A endocytosis, B osmosis, C active transport, Y ATP pumps, and D pinocytosis. So osmosis is the correct answer. Next, which of the following statement is true regarding receptor ligand interaction? A ligand bind to the receptor and trigger the response within the cell. B receptor are located only in the surface of plasma membrane. C receptor ligand interaction do not lead to any change in cellular activity. And D ligand never dissociate from the receptor once bound. So ligand bind to the receptor and trigger the response within the cell. So DNA and RNA questions. Which of the following is responsible for unwinding the double helical during the DNA replication? A DNA ligase, B helicase, topoisomerase, and D primase. So helicase is the correct answer. Next, in eukaryotics, which of the following enzyme is responsible for synthesizing the RNA transcript during the transcription? A. DNA polymerase third, RNA polymerase second, C. DNA polymerase 1, and D. primase. So B is the correct answer. Next, which of the following is a post-transcriptional modification in eukaryotic cell? A. Methylations of cytosine. B. Addition of 5 cap of 3 poly A tail. C. Reverse transcription. D. Replication of DNA. So addition of 5 cap and 3 poly A tail. Next, A transition. The process translation is the process by which a codon mRNA is matched with its corresponding anticodon in tRNA is called A. Transcription, B. Translation, C. Replication, and D. Translocation. So B is the correct answer. Next, which of the following is the primary function of DNA polymerase during the DNA replication? A. Unwinding the double helical. B. Synthesizing the RNA primer. C. Synthesizing the new DNA strand, and D. Adding a 5 cap to mRNA. So C is the correct answer, synthesizing the new DNA strand. Next, which of the following is a type of DNA repair mechanism that correct thymine dimer caused by UV radiation? A. Base excision repair. B. Nucleotide exclusion repair. C. Mismatch repair, and D. Homologous recombination. B is the correct answer. Which of the following techniques is used to amplify DNA sequence? So A is the polymerase chain reaction, B is the southern blotting, C is the western blotting, and D is the ELISA. So ELISA is detect HIV/AIDS, southern blotting, specific protein, western blotting, DNA, but the PCR technology is amplified DNA sequencing. So A is the correct answer. Next, which of the following is a method used for gene editing use a specific enzyme to the target and modify DNA sequence? A. CRISPR's Cas9 PCR, gel electrophoresis, northern blotting. A is the correct answer. Immunity system means it can protect the foreign particle. First question, which of the following is the characteristics of innate immunity system? A. It provides specific response to the pathogen. B. It involves memory cell for faster response upon re-exposure. It provides non-specific response to the pathogen. It requires prior exposure to the pathogen to generate an immune response. It provides non-specific response to pathogen is the correct answer. Innate, inborn immunity. Next, which of the following cell is responsible for production of antibodies in the adaptive immune response? A. T cell, B. Macrophages, C. B cell, and D. Neutrophils. So B cell is the production of adaptive immune response. B cell is plasma cell generate antibodies, and T cell is cellular immunity. Next, what is the primary function of T cell receptor? A. To recognize specific antigen bound to the MSC molecule. B. To produce antibodies against pathogens. C. To activate beta cells in the response to pathogens. D. To stimulate the production of cytokinins. A. To recognize specific antigen bound to the MSC molecule. Next, which of the following describe the major histocompatibility complex MSC molecule? A. MSC molecule help to production of antibodies by the beta cell. MSC molecule present in antigen to T cell. MSC molecule produce cytokinins to activate the immune response, and MSC molecule directly kill pathogens. B. MSC molecule present antigen to T cell. Next, which of the following is true about passive immunity? A. Is acquired through the exposure pathogen. B. It involves the production of memory cell. C. It involves the transfer of antibodies from the external source, and D. It provides long-term protection. So it involves the transfer of antibodies from the external source. Next, which of the following immunological techniques is used to detect and measure concentration of antibodies in antigen? Concentration of antibodies. A. Western blotting, immunodiffusion, C. PCR, and D. Flow cytometry. immunodiffusion. Next, which techniques is used the principle of antigen antibody binding quantify the presence of specific antigen? ELISA. ELISA is the principle of antigen antibody detection. So E is the correct answer. So next, microbiology syllabus. Question is asked, that is, which of the following bacterial species can cause a typical pneumonia? Question is, which one of the following bacterial species can cause a typical pneumonia? A. Chlamydia pneumoniae. B. Streptococcus pneumoniae, Klebsiella pneumonia, and D. Haemophilus influenza. So this is the easiest answer. So that is A is the correct answer. Next, which one of the following organism has axile filaments? A. Mycobacterium tuberculosis. B. Pasteurella multicida. C. Treponema pallidum, and D. Shigella dysentery. So Mycobacterium tuberculosis, this is a TB, so it is cancelled. So C is the correct answer. Next, who among the following scientists was the pioneer in development of chemotherapy? Paul Ehrlich is the correct answer. C is the correct answer. Next, which of the following process glutaraldehyde is used as a sterilizing agent? A. Pasteurization. B. Incineration. C. Cold sterilization, and D. autoclaving. So cold sterilization. Next, the most abundant large quantity class of immunoglobulins in serum is A. IgM, B. IgA, C. IgD, and D. IgG. So immunoglobulins, antibodies to protect from foreign particles. IgG is across IgG. Question is that most abundant large quantity class of immunoglobulins in a serum. So first primary response, that is IgM. IgG is the large quantity available. So D is the correct answer. Next, historical perspective. Who is credited with the discovery of microbial world through the use of microscope? A. Louis Pasteur. B. Robert Koch, Antony van Leeuwenhoek, Edward Jenner. C is the correct answer. Next, which scientist demonstrated that spontaneous generation does not occur through the famous swan neck flask experiment? A. Robert Hooke. B. Louis Pasteur. C. Charles Darwin. D. John Tyndall. Next, the role of microorganism in the causation of disease was first demonstrated by Robert Koch. Edward Jenner developed a vaccine. Alexander Fleming, penicillin. Next, which of the following is a process that involves microorganism in the transformation of organic matter? Fermentation. Next, what is the main purpose of strict plate method in microbiology? Option A, to count the microbial colonies. B, to isolate pure culture. Option C, observe microbial mortality. Option D, to identify microbial species. So option answer is the B is the correct, to isolate the pure culture. Next, second, in microbial nutrition, which of the following is considered an essential nutrient for most microorganisms? A. Glucose, B. Oxygen, C. Nitrogen, and D. Vitamin C. So C is the correct answer. Next, primary purpose of PCR, polymer chain reaction in microbiology, is to A. Isolate microorganism, B. Quantify microbial population, C. Amplify specific DNA sequence, and D. Visualize microorganism under microscope. So amplify the polymer chain reaction role, that is amplify the specific DNA sequence. Next, which sequence technology is primarily used for high throughput sequencing in microbiology? A. Sanger sequencing. B. Next generation sequencing. C. PCR sequencing. And D. Southern blotting. So next generation sequencing. Next, which of the following organism is classified by eukaryotic microbe? A. Bacteria. B. Yeast. C. Archaea. And D. Virus. So B is the correct answer. Next, the classification of virus is based on primarily which of the following? A. Shape of the capsid. B. Type of nucleic acid. C. Size of virus particle. D. Mode of reproduction. So type of nucleic acid. Next, which of the following is true regarding classification of Archaea? A. The share more characteristics with bacteria than with eukaryotes. Next, molecular approaches to the microbial taxonomy primarily rely on analysis which of the following? DNA and RNA sequence. Next, which components of prokaryotic cell is responsible for maintaining its shape and preventing osmotic lysis? A. Plasma membrane. B. Ribosome. C. Cell wall. And D. Flagella. Cell wall. Next, which of the following is involved the movement of bacteria towards or away from the chemical stimuli? Chemotaxis. A. Flagella. B. Ribosome. C. Pili. And D. Capsule. So A is the correct answer. Next, which of the following is the structure is most commonly associated with bacterial adherence to surface? A. Flagella. B. Pili. C. Endospores. And gas vesicle. B is the correct answer. Next, which of the following describe the process by which bacteria can transport a solute across the membrane without using energy? Without using energy. So facilitated diffusion. B is the correct answer. A is the active transport required energy. Next, endospore in bacteria are primarily formed in response to lack of nutrients. Next, primary phase of microbial growth during the which cell divide into constant rate, exponential phase. Next, which of the following method is used to measure microbial growth by counting the number of cell in sample? Plate count method. And C. Direct microscopic count. Next, synchronous growth refers to growth where all cells are in the same phase of the cell cycle. Next, control of microorganism. Which of the following is not a method of sterilization? Autoclaving, pasteurization, dry heat, filtration. So filtration is not or also pasteurization. B is the correct answer. Pasteurization is used for disinfection, not sterilization. Next, which of the following is an effective method for assessing efficacy of disinfectant? Disk diffusion method. Next, which of the following methods microbial control involves the use of ultraviolet radiation? Microbial metabolism. Which of the following pathway is used for microorganism to oxidize glucose anaerobically, means absence of oxygen? That is glycolysis. Next, which electron carrier is involved both aerobic respiration and photosynthesis? Next, interdendroid pathway for alternative, which major metabolic pathway for glucose breakdown? Glycolysis. Which of the following pathway is responsible for synthesis of fatty acid in microorganism? Fatty acid biosynthetic pathway. Next, what is the main function of major histocompatibility complex? Chemotherapeutic antibodies, antibiotics, not antibodies. Which of the following classes of antibiotics inhibit bacterial protein synthesis? Aminoglycosides. Next, which of the following antibiotics is effective against fungal infection? A. Penicillin, B. ciprofloxacin, C. amphotericin, and D. rifampicin. Amphotericin B. Next, which of the following mutation result in change in codon that specific an amino acid? Missense mutation. Next, last, that is general aptitude, 15 marks. In that, a division, that is general aptitude, 5 questions carrying 1 marks and 5 questions carrying 2 marks. So in this sequence 6, 9, 14, and then what is the x value? 30 and 41. Possible value is 6, 9, 14. So we have observed just general aptitude in that in the sequence of 6, 9, 14, x 13 and 41. So here we observe 6 in plus 3, that is 9, 14 and 14 into how much? 9 plus 14 how much? So increase value, that is 5. These two have difference. Next, 9 plus 14. 14 plus how much? Option A is that 25, B 21, C 18 and D 20. So 21 is the correct answer. Next, a person sold 2 different items at the same price. He made 10% profit in one item and 10% loss in other item. In the selling those 2 items, in the person made total of A 1% profit, B 2% profit, C 1% loss, and D 2% loss. 1% loss. Next, a cube is to be cut into 8 pieces equal to size and shape. Here each cut should be a straight and it should be not stop till reaches at other end of the cube. The minimum number of such cut is required. 3. Next, an engineering college of 10,000 students and 15 like neither their core branches nor other branches. The number of students who like their core branches is 1/4 of the number of students who like other branches. The number of students who like both their core and other branches is 500. The number of students who like their core branches. To wizard. Next, which of the following is the correct form of verb in the sentence? See dash dash to the market every day. See go, goes, gone, and going. She goes. Choose the correct sentence. She have a dog. She has a dog. She having a dog. She had a dog. Next, which of the following is an Adams? Bite to the bullet. Be large dog. Fast car. Red apple. Bite the bullet. So A is the correct answer. Next, identify the preposition of sentence. The cat is under the table. Preposition cat under table is. So under. B is the correct answer. Next, quantitative aptitude. So I have to practice and solve these problems. Spatial aptitude, ask the questions. Data interpretations. So this examination, some questions are difficult, but some questions are easy. To try to focus, those are easy, and then those are difficult. So to develop the strength, and this is a very good opportunity. Because stipend is that 12,400. So need solve the previous year question paper 3-4 years, as well as prepare the notes, read, revise consistently. Best of luck your preparation and examination. Hope this video is helpful for your examination. Thank you so much for taking the time to watch this video. We truly appreciate your support and hope you found the lecture helpful. And all the best for your examination.